Shri Gurubhyo Namaha. Welcome to our online teaching academy Ekalaiva, and I welcome you all for the science class. This is class 10 science. Here we are going to see about biology part. In biology we have different lessons like life process, how do organisms reproduce. Now we are going to step into new lesson, and it's totally new for 10th standard, it's basic for 12th also. Here I'll show you a picture, just guess what is that picture. We'll see the lesson name, it's Heredity and Evolution. Mr. G.J. Mendel, we call him as father of genetics. Yeah, the picture is on here. Check the picture. I think it's ruling the world, this called virus. And you all know the name of this virus, coronavirus or COVID-19. So what happened? It is a type of virus which you call it a SARS virus. It belongs to SARS type. And this virus, some 20 years back, some few decades back, what happened? It was not lethal. It was like a normal virus, not infecting us. But now, this has transformed to something because of one particular genetic material. This virus, actually we call it as RNA virus. Okay, this RNA got triggered. It got evoluted because of mutation. Okay, we can say like this, it got mutated. Because of that, it has become very lethal and it's highly infectious now. Okay, as I said at the beginning, it's ruling the world. Okay, so we are going to talk about this genetic material, the gene. So this lesson totally about the gene. We call this unit as genetics. Here one topic alone we are going to see, heredity. What is heredity? The character, the transmission of character from one generation to another generation. So, before going into the lesson, we will see some introduction about what is genetics. So, what is genetics? It is a branch of biology. It's a branch of biology that deals with the heredity, especially the mechanism of hereditary transmission, the transmission of the characters from one generation to the other generation, and the variation. So what is variation? Something changes, the change in the characters, we can say. Everyone are not same. Okay, there are some changes. If you take from asexually reproducing organism to sexually reproducing organism, there are variation. In how do organism lesson, we read already, asexually reproducing organism will have less variation, whereas sexually reproducing organism will have more variation. So we are going to see about that also. Here about bacteria we will be seeing later. Mendelian genetics, in the beginning I told you who is the father of genetics, G.J. Mendel or Gregor John Mendel. Okay, about Mendel's history, who he was. He was a monk, okay, working in a monastery, and then he was a teacher also, but not a great teacher, but he was very much interested in training, that is mathematics, statistics, probability. So he did a research work. In his research he incorporated all these subjects, mathematics and probability. If you see Mendel's research work, mostly will be dealing with some calculation also, and he got experimental evidence for all those things, and it's worldwide accepted. His success based on the two important things. Okay, Mendel selected an experimental species called Pisum sativum. We call it as pea plant in general term. Why you want to select that particular species? What is so great in the species? Anything experimentally you want to select, okay, that species should have shorter lifespan. Then only we can do more experiment and we can get more result in shorter period of time. Okay, that's why he selected this Pisum sativum. So he used a garden pea plant for test subject. Okay, it's test subject, garden pea plant. Why he want to select this pea plant? Because of 6 reasons. I told you already, it is easy to grow, very short lifespan, it is small, produce many offsprings, mature quickly, more varieties we have. When it is variety, we are going to talk about contrasting characters, contrasting traits or characters. Some 7 contrasting characters we are going to talk here. easy to fertilize, okay, self-fertilization as well as cross-fertilization. We know that self-fertilization or self-pollination in the same plant, both the male flower and the female flower, or the male part or the female part will be there in the same flower. So, it is self-fertilization. Cross-pollination or cross-fertilization, two different. So, this plant is adaptable in both the condition. So, this is another advantage. He used a quantitative approach, quantity, number. When you are conducting an experiment, the number, how much you take, that is very important. Suppose if you are going to take only 100 number and going to get a result, next time if you are going to take 1000 or 10,000, you may not get the same number. So, Mendel was so clever and he selected the species in large number, humongous number, and he got a good result, but his work was not recognized when he was alive. But later, some 3 scientists, they came and rediscovered about Mendelian work, and now it all came into light. So, that is it about the Mendel and why Mendel was successful also. Now, we will see about the plant. This part is very important for 10th standard, and that too you will get board based question from 7 contrasting characters, and this is the basic. From here only we will be talking about the cross. Later in this lesson we will be talking about the different types of cross, monohybrid and dihybrid. That is the main theme of the lesson, I can say. Mostly questions will be arise from that particular part only. Before that, we should know the basic knowledge of the pea plant. So, if it is a pea plant, a plant will have a root, stem, flower, leaf, etc. But what character is selected? Mendel selected 7 contrasting character, and that character, how it is going to get transmitted from one generation to another generation. Okay, that is very important. That you call it as heredity, the transmission of the character. He was observing that point and then checking what are the characters he is going to get in the next generation, and the generation also he calculated as first generation, second generation, like that, and how many in number. So, here so many terminologies we are going to explore. So, listen carefully. First, plant will take it, pea plant, Pisum sativum. So, in the pea plant, what are the things? You can see the parts of the plant here. Yes, very well, you can see the parts of the plant. So, first we have taken the seeds, then the flower color, then pod, form as well as the color, then stem, position of inflorescence, nothing but flower, and then size. So, one by one we will see in detail. First, seed form, otherwise called as shape of the seed. So, round, okay, we will use any one term, round, wrinkled. Then cotyledon, if you take yellow color, green color. Now, what we are learning here? We are learning a term, one term I am going to say. So, listen carefully, it is a phenotype. We are talking about the physical appearance. When you look a plant, you say the plant is tall, the plant is short. Okay, you can say the color of the flower is red and the color of the flower is white. So, what you are seeing, the physical appearance, and that you call it as phenotype. Okay. So, here we are talking about the phenotype. Okay, when I say round or yellow or the shape, whatever it is, that is a physical character, phenotype. And now we are saying the form, cotyledon color, flower color, pod color, that you call it as traits, contrasting traits or the contrasting characters. Okay. So, phenotypes we have 1, 2, 3, 4, 5, 6, 7 different phenotypes. That is why in the beginning I said 7 contrasting characters. So, next one, white flower, violet or red. In some book it will be in purple. Okay, some book they would have given purple color also. We will follow our NCERT, violet. Okay, then it is full, the form, again the form is nothing but shape. It is full, constricted, and color, if you take it yellow color, green color. This is color of the pod. Okay, you can see the pod shape as well as the color. Constricted, you can see how it is got constricted like this, right? Then position of the inflorescence, flower position. There is a difference in flower position, right? One is axial in position, another one terminal in position. Terminal, okay, you know, 2 terminal, positive and negative. So, it is at nothing but end, like that. Terminal is nothing but end here. So, the flower position in terminal and the flower position in axial. Then next you can see the size, the long you can call it as tall, and then this you can call it as dwarf. Size of the plant. Okay. Now, in next slide we are going to see the dominant and recessive. In that I am going to tell you the genotype also. Here two things we discussed. One, the 7 contrasting characters, and next we discussed about the phenotype, the physical appearance. Next we are going to talk about the genotype. Okay. So this area, another important terminologies we are going to discuss. So listen, here new term you can see, dominant and recessive. So what is dominant? When the word comes dominant, only that character will be seen. Okay, he is dominating. Okay, so only that particular person's wish we are taking, that's it. Okay, that is called dominance. Like that, here the characters are dominating, not allowing the other character to show their presence or the appearance. That's why you call it as dominant. So what are the characters? How are we going to represent their gene? Now we are traveling into the gene level. So far we were talking about the physical appearance, how they look, that's it. Now the lesson is about heredity. Mendel was discussing about the gene. So you want to represent the character letter gene. So how to represent the letter? We are going to take the dominant characters first letter and we are going to write in a pair, capital letter. Okay. So we will see here. We are dividing into two characters, dominant and recessive. Later I will tell you why we are calling it as dominant. We discussed dominant means it will be dominant, it will show their presence. Recessive cannot show. Okay. So we will see about genotype, then still more in detail we can discuss. Genotype. Already we wrote the phenotype. Now genotype. In this, which is dominant? This is dominant, purple. So first letter you will take it and that you will represent in capital letter as allele. Okay, we call this gene which is occurring in a pair or called allele. For example, in chromosome. Okay, chromosomes all occur in pair only, right? Chromosomes all will occur in pair. And in this each chromosome you will have that character. Suppose if I say purple color, one character here, okay, one P here, another P here, and this you call it as alleles. Okay, in simple term genotype you can write it. So this is PP, capital letter we are using, and it's nothing but genotype. The same here, recessive character, you will represent by small p. Okay, the same P you will take it and that you will use as capital letter for dominant and the same letter you will use as small, lowercase for recessive character. Now we will write for the other thing. Plant height, tall, tall is dominant. So capital T, capital T. Same what you will write here, small t, small t. Yellow, seed color. Okay, these two areas you need to concentrate. One is seed color, yellow is dominant, but pod color, green is dominant. Okay, so this Y in lower case you will write for recessive. The same thing, seed shape, round, capital R, capital R. The same you can write for wrinkle, same letter. You will not have any confusion if you are thorough in this basic concept. Same letter, uppercase, lowercase. Uppercase dominant, lowercase recessive. And here small g, small g. Then inflator, full. So I, capital. Small I, recessive. Axial, dominant. So capital A, capital A. Terminal, small a, small a. I am not using any of their starting letter because they are all recessive character. So this is a way to represent the genotype. So two terminology we know very well. One is phenotype, another one genotype. And next also we discussed dominant, recessive. In a very simple term I will tell you. Two plants. One is tall plant, another one dwarf plant. Two plants they are going to intercross. Okay, Mendel did the intercross between these two plants by pollination, bagging method. Then what happened? The next generation he got a tall plant. When he intercrossed these two, he is getting a tall plant. From that only he came to know, okay, the other character suppressed, masked, okay, not allowing to express. Only the dominant characters were allowed to express. Like that he did research for all these characters and got the dominance as well as recessive. Okay. So the same thing, we will go terminology still more in deeper, the other terminologies. Gene. Gene is the functional unit. Okay, it is a functional unit of inheritance. the gene. Okay. Then the gene word was coined by Johansen, and it was called as factor by Mendel. Okay, later it was corrected as gene. Next I told you about allele. Okay, what are the allele? The dominant. For example, tall, for tall you will write capital T, capital T. This is one allele. Okay, and recessive, small t, small t. This is another allele. Okay, like this we are representing with the two letters in a pair. When they occur in pair only, they can show their character. This part you have to keep in mind. It will be helpful for doing the cross. Then next, multiple allele. We know this one only. For example, if you take blood group, okay, there are multiple. Some may be A or B or O or AB. In that case, we can say multiple alleles. Even that part we will see in blood grouping. Okay. Next we discussed about phenotype and genotype. Okay, just now we discussed physical appearance, it is the genetic makeup. Again we will write one example. Tall is the phenotype. Genotype we are representing by the letter capital T, capital T. What are chromosomes? Okay, where are the gene present? Again, if this is a cell, in this cell there is a nucleus. Inside the nucleus there is nucleolus, and here there is a thread like coiled structure, you call it as chromosome. So from chromosome, actually you call it as chromatid, and this chromatid during cell division they become condensed. That time you call it as chromosome. So it will look like this in a pair. We call this as homologous chromosome, and then this is chromosome. Inside the chromosome there is again thread like structure, and that thread like structure if you pull out and see, it is nothing but DNA. What is DNA? DNA is deoxyribonucleic acid, and in the DNA you can see the genes. So we have pulled out the gene from the cell, right? So this is a chromosome, a rod shaped structure within the cell nucleus that carry genes encoded by DNA. You can see all the parts here, right? DNA, still more in detail, it is deoxyribonucleic acid. Okay, this expansion is very important. Then composed of nucleic acid. What are the nucleic acid? Deoxyribonucleic acid. These molecules encode the gene that allow the genetic material or information to be passed into the offspring. We are saying no variation, some inheritance pattern. Father will be having curly hair, even the child will be having the curly hair. How it is getting transmitted? It's all because of gene, inheritance. And then mutation. In the beginning we spoke about COVID. Okay, how this COVID has transformed totally and infecting because it underwent mutation. Okay, so sudden change in the gene. Next, another important term. Yeah, homozygous, heterozygous. These terms are very important. I think when I write like this, it will be clear for you. This already we know it represent female. XY represent male. Already would have read in 8th standard. The same thing if I am going to write capital T, which is nothing but tall, small t, okay, which is nothing but dwarf. Okay, this small t also homozygous, and the same thing when I represent like this capital T and small t, it is heterozygous. I think from this you can figure out what is heterozygous. Two different letters, okay, when they are coming, or two different things, lowercase and uppercase, you call that heterozygous because it is having two different characters. Okay, and homozygous the same. Let's say homo, it is homozygous. Locus, if this is the chromosome, imagine if this is the chromosome and here are the gene, we can say as the position 1, 2, 3, we are locating the gene, right? That part you call it as locus. The location of the allelic gene on the chromosome, you call it as locus. Totally we have completed the terminologies. In next class we are going to see everything in detail applying these terminology like monohybrid cross and dihybrid cross. To receive our online lessons, please press the subscribe button and you will receive the latest update. Thank you.